Hello everyone. In this video, I'm going to explain how transcription and gene expression is happening. You should know that this section is for higher level and it is from 7.2. You will see in this slide the structure of RNA polymerase and the direction of the 5' to 3' for making new RNA, which is called mRNA. And there are so many other complex and proteins over here. There are so many interesting things happening over here. The place that will be recognized by the RNA polymerase, the place that will enhance or silence the amount of the transcription, and which have different names, and you will learn all about this area later at this section. The essential idea is that the information from DNA will be used to create mRNA. Information stored as a code in the DNA is copied into mRNA. So the purpose of making mRNA is transferring the information which was stored in DNA, and that information will be translated into protein, which is necessary for other functions. So you can see over here that in transcription, a portion of the double strand DNA template gives rise to a single-stranded RNA molecules, and these parts are having genetic code, and the mRNA transfers these genetic codes in and go to the translation machine in order to make a protein or enzyme depending on what the cell needs. Some of the things over here in this table you have learned it before. For example, you already know the transcription direction is from 5' to 3'. You know the role of nucleosomes. So we had this conversation and discussion in the previous slide and the videos. And you also learn about some modification of the mRNA, but maybe not very specific and in detail. So we are going to learn more on the splicing, which is happening on the mRNA. So splicing of mRNA increases the number of different proteins an organism can produce. This is something that we didn't discuss it before. The role of splicing and how can splicing increase the number of the different of the proteins, and the gene expression is regulated by proteins that bind to specific base sequences in DNA. This gene regulation, this gene expression and regulation is something that we will discuss it very nicely and with more details. And I love these last three. I love these. That's so interesting. The environment of a cell and of an organism has an impact on gene expression. Can you imagine that? That the environment, by changing the gene expression, can change the genetic, but that is not really genetic. It is called epigenetic. I love this topic. Okay, so we will have a full conversation and discussion on epigenetic. The promoter as an example of non-coding DNA with the function, analysis of changes in the DNA methylation patterns. So we are talking about how this epigenetic is functioning, how does this work, what does the methylation means, and the function of the promoter, which is a kind of non-coding DNA, and its function. So these are the things that we are going to learn at this section, and it is so exciting and interesting. This area we have already discussed about, and if you go back to the previous video, you will find the information and exactly the same slide. It is about the exon and introns, and each gene includes in the DNA, including exon and intron. And if you remember, I told you that exon are extremely important, so will be kept, but introns will be removed. And also we have some enhancer and silencers before each gene, which have the effect on the amount of the transcription and the copies coming out from that gene. If you remember, when we talked about non-coding region function, we mentioned many times related to those non-coding regions which are used for finding the similarities between people as tandem repeats, and the role that they had in telomeres at the end of the chromosomes for protecting the chromosomes. But one of the most important non-coding regions in the DNA, which is not translated but is very important and has a very important function that we didn't mention at that slide, and beyond that too, is promoter. What is the function of promoter? Promoter is some area located before the gene, and it is the binding site, which means that it will be recognized by the RNA polymerase. So it is very important because this is the place that RNA polymerase is coming and recognizing and start to do the transcription after finding the promoter. After promoter, we have other two areas. The other one is operator, and the other one is the exact gene. So the operator, we had some discussion on operator as a form of silencer or enhancer. Operator is an area that is the DNA, is the region of the DNA that regulates transcription. It has the function on transcription regulation, and this regulation could be by enhancing or silencing. So it could inhibit the transcription as a silencer, or it could increase the transcription as an enhancer. And we had this conversation before. So here is a very common and famous example of how a gene is working, and this is called lac operon. So if you Google lac operon, and if you find this word, millions of searches related to this is coming up, and you will see what does that mean. So lac operon is lac. Do you know what does lac means? And lac comes from what? You can guess, I think. So lac is coming from lactose because it is related to lactose, and lac operon is something which is operating related to lactose. And you know that lactose is a sugar, and this sugar is inside the milk, and this lactose should be broken down unless you don't have the enzyme for breaking down the lactose. So a study about the lactose breaking down by the enzyme was very important, and the function is based on how this gene works. Okay, what you need to know about the lac operon is that, first of all, what is the function of this gene by producing the protein which has the function of breaking down the lactose. First, what is the name of that protein? That protein or enzyme is lactase. So you see that the enzyme which is breaking down the lactose is lactase. So this gene is producing the enzyme which is called lactase, and it has the function on breaking down the lactose sugar. How this gene is regulated? The regulation of this gene is controlled by a repressor protein, and this repressor protein is bound to the operator. And once the repressor is bound to the operator, it doesn't allow the gene expression happening. Here is the place of RNA polymerase, which recognize already the promoter, and it's ready for doing the gene expression, but there is something doesn't allow, and that's the repressor. So repressor binding to the operator, and it doesn't allow the gene expression happening, so there is no transcription over here. This is exactly the same as the example that we had on silencer example. So if in this case we have a silencer attached to our operator, but we don't call it silencer here, we call it repressor protein. Okay, let's get back to this, the meaning of really why repressor is binding to operator. The reason is there is no lactose. When there is no lactose, when there is no sugar, then it is not necessary to produce lactase. So whenever we don't have lactose, we don't need lactase. That's so simple. So this repressor is regulating that. And then when the cell is noticing that there is a lactose and we need lactase. Okay, yeah, let's look at the next slide, and then you will see that how the cell notices about the bacterium, actually the E. coli bacterium notices about the presence of lactose. Once there is a lactose in the environment, then this repressor is binding to that lactose. So in a regular way, at the time that there is no lactose, this repressor already attached here, and it doesn't allow the expression of the gene. But once there is lactose, when there are lactose molecules over here, and you see that the lactose molecules are around, and then this repressor not necessarily attached to operator anymore. So it starts to bind to lactose molecule. And then what is the consequence of binding this repressor to this lactose molecule? Then the consequence is that it will free the area which is operator, and then it will give the permission, and it makes it easy or make it free that the RNA polymerase that has already been attached to the promoter over here, then it starts to do the transcription. So that's really nice and simple, the mechanism of lac operon. The function of the enzyme after production and after transcription from the gene has not clearly drawn over here, but the function is something that has been written over here. This means that once the lactase will be produced after transcription of the gene, then this lactase can come and break down these lactose molecule. So lactose will be broken down after the lactase will be produced, and the lactase will break down this sugar because it is important for us for digestion that the lactose will be broken down. So this is a summary of the common types of regulating proteins and associated sequences found in eukaryotes. We have enhancer as the role of the enhancer. Enhancers are activating. We have silencer, and the role is as repressor. So this is what we just studied related to lac Operon in E. coli. And the area which is called promoter, and it is for attachment of the RNA polymerase.
The E. coli lac Operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac Operon: beta-galactosidase and permease. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac Operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac Operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac Operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.